Hey everybody, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania, which is just outside of Philadelphia. Again, I teach A&P at Bucks County Community College, and I've been doing that since 2002. In today's episode, we're going to move on with the blood, and we're going to specifically talk about leukocytes and platelets, which is basically white blood cells and cell fragments that we call platelets. We're going to get into how white blood cells form, which is called leukopoiesis, the different kinds of white blood cells we have, and what each of those different kinds does in a very general kind of way. Since the white blood cells are very much active in immunity, we're going to get deeper into them when we get to the immune system. And then we'll see a little bit more in-depth discussion of what some of these white blood cells do. When it comes to platelets, we're going to talk mostly about what we call hemostasis, which is the cessation of bleeding. So if you have an injury where there is an open blood vessel, we need to make sure that you don't lose too much blood. And we do that through a few processes that contribute to what's called hemostasis. So we're basically protecting against blood loss. We get a lot of content in this episode, so I think we're going to get right into it. This episode goes a little bit long because I wanted to finish up to talk about blood cells. So that'll take us through this entire episode. So without any further ado, let's start with leukocytes. All right, so leukocytes are commonly known as white blood cells. Their production begins in the red bone marrow, and we call the production of leukocytes leukopoiesis. Similar to erythropoiesis is for erythrocytes, and they both begin in the red bone marrow. When it comes to leukocytes, their primary function is immune function. So basically our immune system's response to injury, infection, and disease. Unlike erythrocytes or red blood cells, leukocytes leave the circulatory system's vessels on a regular basis as part of their normal function. In fact, at any given time, there are more leukocytes in our interstitial fluid than in our blood vessels. In between the squamous cells of a capillary, which is a microscopic blood vessel, there are intercellular clefts, right? So that's a space between two cells. And those basically are small gaps that allow leukocytes, which are pretty flexible, to squeeze through them in a process called diapedesis. Leukocytes are drawn to sites of injury or infection by chemicals released from dead or injured cells, as well as pathogens, in a process known as chemotaxis. The dilation of the blood vessels in response to injury or infection helps facilitate diapedesis and the filtration of blood plasma into the interstitial fluid. This is one of the main non-specific immune responses, and you've probably already figured out that it's called inflammation. So when you have an injury or an infection, those pathogens or those injured cells are going to release chemicals, and those chemicals are going to draw blood plasma and white blood cells into the area that's injured or infected. This is the swelling that takes place. That dilated blood vessel that draws more fluid in is going to cause those tissues to swell or inflame. That is a non-specific immune response to help fight the infection or to help heal the injury, so to get rid of the dead and damaged tissues. This inflammation is, again, a non-specific form of immunity. It causes pain because distension of tissues can trigger nociceptors. It can also cause itchiness because localized inflammation can result in the perception of the itch sensation. Now, leukocytes are divided into two categories based on whether or not they include specific granules in their cytosol. Specific granules are secretory vesicles that contain enzymes or cytotoxic chemicals that function to create immune responses. One example of a cytotoxic chemical is lysozyme. Lysozyme has antimicrobial properties that can break down the cell wall of a bacterium to protect us against infection. So cytotoxic chemicals means chemicals that can destroy a cell, specifically a bacterium, which would be nice, or a damaged cell or a dead cell that we want to break down and get rid of. The leukocytes that have these specific granules are called granulocytes, and that includes neutrophils, eosinophils, and basophils. These are 3 kinds of leukocytes. Those that lack specific granules are called agranulocytes, and that includes lymphocytes and monocytes. Each leukocyte has a unique structure and function. So let's back up for a second because I mentioned something here that I want to just give you an intro to. And that is that leukocytes have their own specific classes, right? So we've got granulocytes and agranulocytes. And in those classes of leukocytes, we have specific leukocytes. One is called a neutrophil. Then we have basophils, eosinophils, lymphocytes, and monocytes. And we're even going to break down some of those in more specifics. For example, we have T lymphocytes and B lymphocytes. And monocytes are sometimes going to differentiate into macrophages. So we're going to talk a lot about that as we move forward, and especially as we get into talking about our immune system in more detail later on. Each of these types of leukocyte has their own specific structure and function. All right, let's get into leukopoiesis a little bit. So like I said before, it takes place in the red bone marrow, and the original stem cells are the same hemopoietic stem cells that give rise to erythrocytes. Some of these hemopoietic stem cells can differentiate into distinct types of colony-forming units, or CFUs, one for each of the 5 types of leukocyte. The CFUs are stimulated by chemical messengers called colony-stimulating factors, or CSFs. Each CFU develops into a more specific stem cell called a blast cell. Blast is a suffix that typically means that the cell is not quite mature yet. It is still working on it. So when you hear something like osteoblast, you've heard that before in previous episodes, or chondroblast, that usually means that that cell is not fully mature yet. So keep that in mind. All right, let's talk about the granulocytes for a second. Each of them is going to form into its own specific myeloblast, and the agranulocytes form from monoblasts and lymphoblasts. The development of the granulocytes all follow the same pattern. The myeloblasts become promyelocytes, and the promyelocytes become myelocytes. That prefix pro typically means that that cell is an immediate precursor to another cell. So a promyelocyte is going to become a myelocyte as it matures. So there are eosinophilic, basophilic, and neutrophilic versions of each of these stages of development, which then respectively differentiate into eosinophils, basophils, and neutrophils. Monoblasts give rise to promonocytes, which then differentiate into mature monocytes. Lymphoblasts can give rise to 3 different prolymphocytes, a B, a T, and a natural killer prolymphocyte, which then differentiate into B lymphocytes, T lymphocytes, and natural killer cells, respectively. So it makes sense, right? You think about that term, prolymphocyte, which means it's going to become a lymphocyte, and we specifically have a B, a T, and a natural killer, so those are going to develop into those kinds of cells. And we're going to talk about what all these different cells do a little bit later on in the episode. The colony-stimulating factors that facilitate the development of leukocytes are specific to the kind of leukocyte needed by any particular situation in the body. For example, a bacterial infection may result in the CSFs that target the development of neutrophils, which function to destroy pathogens. They may also result from allergic reactions, which could then stimulate the development of eosinophils, because eosinophils function to combat allergens. Granulocytes, that's the neutrophils, the basophils, the eosinophils, and monocytes are stored in the red bone marrow until a situation that requires them arises in your body. There can be up to 20 times more leukocytes in the red bone marrow than in the circulation. Now that leaves us with lymphocytes, which are the only ones that do things a little bit differently. Lymphocytes leave the bone marrow and make their way to the thymus to complete their development. From there, they colonize in lymphatic tissue, and can survive for decades, cycling through the lymphatic system, the systemic tissues, and the bloodstream over and over and over again. So what is lymphatic tissue? So let's talk about that for a little bit. You've probably heard the term lymph. Lymph is a fluid in your body in vessels. When your blood capillaries course through your tissues and deliver blood plasma and its solutes and pick up interstitial fluid and its solutes, that's a good system for exchange. So we have capillary exchange. We deliver stuff to our tissues and we pick stuff up from our tissues. Only about 85% or so of the fluid that leaves the capillary into the interstitial fluid actually returns to that capillary to go back into your circulatory system. That means that we've got extra fluid in your tissues that we have to do something with. Otherwise, we'll just swell up beyond belief. That extra fluid gets absorbed into lymphatic vessels and becomes lymph. And those lymphatic vessels lead to lymphatic tissues. Some examples of lymphatic tissues are your spleen, your thymus, your tonsils, and your lymph nodes. And there's other places as well that we have lymphatic tissue, but we're going to have episodes on the lymphatic system, so we don't have to get too much into it in any more detail there. But those lymphatic tissues are really ripe with lymphocytes, because what they're going to do is they're going to take that fluid and they're going to clean it up. They're going to clean it up. They're going to get rid of any dead cells, damaged cells, viruses, bacteria, anything like that that we don't want circulating in your system. And then they're going to take that lymph and they're going to deposit it back into the circulatory system. All right, now granulocytes, on the other hand, spend a short time in the bloodstream, only lasting between 4 and 8 hours before they filter through the capillary walls and into the tissues. Once they're in the tissues, though, they can live like up to 5 days. Monocytes can last in the blood for up to 20 hours before they end up in the tissues. Then they transform into macrophages, which can survive for a few years. Now let's get back to those lymphocytes that can be in your system for decades. One of the reasons why that's really good is because lymphocytes are the cells that are going to produce antibodies. Antibodies are proteins that recognize and attack antigens on invading cells or viruses. So this way, when you get infected by a virus, let's say it's chickenpox, your lymphocytes eventually, and I'm not going to get into the details of this just yet, but eventually lead to the production of antibodies that can bind to the antigens on those viruses and make it so they can't infect you again. And you can make those antibodies for decades because those cells last for decades in your body. And so that's why you typically don't get chickenpox twice. In fact, there's a lot of viruses that you don't get twice. And that's because those viruses are fairly stable kind of virus that doesn't mutate too much, and because we're pretty good at making those antibodies. Some viruses, like viruses that cause the common cold and the flu, they mutate a lot and their antigens change. So the antibodies that you made for them the first time you got exposed aren't going to work the next year, for example, because that virus has mutated, its antigen has changed, and that antibody can't bind to it anymore. And that's why for some types of vaccines, we get boosters, like a flu shot. Every year, it's a different flu shot because they're anticipating the kind of flu strain that's going to come around, and they make a vaccine for it. Same with COVID. The COVID vaccine changes because COVID is a coronavirus that does a lot of mutation, and the strain changes every year or so. All right, so let's take a look at the leukocytes individually. So you've probably seen a blood test where you get a CBC, or a complete blood count, and that means that you're going to get a result that tells you how many red blood cells, how many white blood cells, and how many platelets. If you see a test that calls it a CBC with a diff, or differential, that means that they're going to differentiate out all the leukocytes. So in that test, you're going to see not only white blood cells, but you're going to see neutrophils, eosinophils, basophils, monocytes, lymphocytes. You might even see something called a PMN, and I'll talk about that in a minute. So let's start talking about the different types of leukocytes, and we will start with the granulocytes. Each of the granulocytes is about 1.5 times the size of an erythrocyte. Neutrophils are by far the kind of leukocyte we have the most of. They make up about 50 to 70% of all white blood cells, or leukocytes. They have granules, but they're faint. So their cytosol has specific granules. They're kind of faint. You can't really see them very well. And they've got an interesting nucleus. Their nucleus is multi-lobed, with big fat parts and then skinny parts connecting another big fat part. There can be anywhere from 3 to 5 lobes to a neutrophil's nucleus. And for that reason, you might see PMN on a lab report. PMN stands for polymorphonucleated cells, which means many-shaped nucleus. Poly for many, morpho for shape, nucleated for nucleus. Neutrophils are sharply elevated in response to a bacterial infection because they function to phagocytize bacteria. Now, remember, phagocytize means to engulf and destroy something. So when a white blood cell has its immune function by engulfing a foreign particle, like a bacterium, and then destroying it from the inside, and then spitting out all of its parts, that's called phagocytosis. In addition to phagocytosis, they also release enzymes that are hydrolytic, and they target pathogens. Hydrolytic means they're going to destroy those pathogens. Eosinophils are pretty rare. They only make up between 1 and 4% of all leukocytes, and their granules have a reddish hue. Each eosinophil's nucleus has 2 lobes, each connected by an isthmus. An isthmus is like the little skinny piece of land that connects 2 islands. Eosinophils assist the immune system by phagocytizing antigen-antibody complexes. So let's talk about that for a second. Earlier I mentioned antibodies and antigens. We talked about antigens in the previous episode where we discussed blood type. Antigens are these glycoproteins usually that are on the surface of a cell or some other kind of organism. And they are identifying. They're unique. When we make antibodies against the antigens of a foreign invader, like a bacterium or a virus, those antibodies will bind to the antigen, and then they will render them harmless against you. That now becomes an antigen-antibody complex. And instead of just letting them float around your body forever, eosinophils grab those up, engulf them, and destroy them. Phagocytosis. So eosinophils assist the immune system by phagocytizing antigen-antibody complexes. They also lessen the allergic response by phagocytizing allergens, which are the antigens that cause allergic reactions, like those associated with pollen or pet dander. Eosinophils also respond to parasitic worm infections. Parasitic worms are roundworms, tapeworms, flatworms. These are some of the more common types of worms that can inhabit the human digestive tract. And those infections can really cause problems for people. So one of the defenses against parasitic worms is eosinophils. So what they'll do is they will release cytotoxic chemicals that can destroy those worms. Basophils are the least common white blood cell we have. Up to 1% max for basophils. They have dark, abundant granules that secrete histamine and heparin. You've probably heard of histamine, or at least you've heard of an antihistamine, which is common in allergy medicines. Histamine is released in response to allergic exposure and causes vasodilation, which in turn makes the capillaries more permeable to blood plasma. That vasodilation results in inflammation of the mucous membranes as the blood plasma filters out of the capillary and into that tissue fluid. The symptoms associated with allergies, like runny nose, itchy red watery eyes, those are caused by the vasodilation. Heparin is an anticoagulant, means it inhibits blood clotting, and it's often prescribed to patients who are at risk for excessive blood clots. Sometimes you'll hear anticoagulants called blood thinners.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times. But we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
All right, now on to the agranulocytes, which are lymphocytes and monocytes. Lymphocytes are about 20 to 40% of the body's leukocytes, and they exist as inactive white blood cells until they encounter a specific antigen and other chemical signals. Inactive lymphocytes are smaller than other leukocytes, measuring roughly the same size as an erythrocyte, and sometimes even a little smaller and sometimes a little larger. They have a darkly staining, proportionally large, round nucleus. Sometimes you can barely make out the cytoplasm in an inactive lymphocyte because the nucleus is so big. Activated lymphocytes grow to about twice the size of an erythrocyte with an increase in the volume of cytosol surrounding the nucleus. Lymphocytes fall into one of 3 categories: T lymphocytes, B lymphocytes, and natural killer cells. Their names are often shortened to T cells, B cells, and NK cells. You've probably heard of T cells. We usually use our T cell count to measure our immune function. T lymphocytes are directly involved in an immune response. Some T cells attack foreign cells, pathogens, and our own cells that have been infected by a virus. The T in T lymphocytes stands for thymus, which is where T cells mature and learn how to identify our own self-antigens so they don't attack them. So we call that process immunocompetence. The T lymphocytes are initially produced in the red bone marrow, and then they make their way to the thymus. The thymus puts them through a series of tests to see if those T lymphocytes can recognize your own self-antigens, which means the antigens that are on your own cells based on your own DNA. These are identifying markers. We want to make sure that our immune system does not attack our own cells. The immune system's job is to attack other cells coming into our body, pathogens, foreign invaders that come into our bodies and could make us sick. What we don't want is our own immune system attacking our own cells. When that happens, we call that an autoimmune disease. Remember, auto means self, like the person who writes an autobiography is the person who it's about. That immunocompetence is achieved in the thymus. And last I heard, about 98% of the T lymphocytes fail the immunocompetency tests and they're destroyed. The remaining 2% are allowed to go back out into the circulatory system and patrol your body for foreign invaders without attacking your own cells. Now, some people think the B in B lymphocytes stands for bone marrow because that's where they were produced. However, the B in B lymphocytes actually refers to a bursa in birds where they were first discovered. In humans, however, all blood cells are produced in the bone marrow. When stimulated by an antigen, like the ones found on viruses and bacteria, B lymphocytes differentiate into a different kind of cell called a plasma cell. And the plasma cells synthesize the specific antibodies that can bind to the antigen of that virus or bacterium and protect us from future infections from it. So remember, it's not the B lymphocytes that directly produce the antibodies. It's the plasma cells that the B lymphocytes differentiate into. Natural killer cells stimulate apoptosis, which is known as programmed cell death. Cells that have been infected by viruses or bacteria or cells that are tumor cells, they have this programmed cell death, and the apoptosis will just allow those cells to destroy themselves because they're infected or they're tumor cells. The natural killer cells stimulate the apoptosis. They also go after the donor tissue of organs that have been transplanted from another individual. So remember, you might get an organ transplant, and that's good. However, you have an immune system that is trained to recognize cells that aren't you. So when that transplanted tissue comes in, your immune system will naturally go after it and attack it. So what we do is we give transplant patients immunosuppressive drugs like cortisone to limit the attack on transplanted tissue. All right, finally, monocytes. Monocytes are another agranulocyte, just like lymphocytes, and they make up somewhere between 2 and 8% of our leukocyte population, and they're the largest of all of our blood cells. They're about 3 times the size of an erythrocyte. Their nuclei are large and crescent-shaped. After about 3 days in the circulation, monocytes filter into the tissue fluids and transform into phagocytes called macrophages. Macrophages engulf and destroy cellular debris, dead cells, cell fragments, bacteria, and viruses. All right, so those are the 5 different kinds of leukocytes and the subsets. Remember, they're broken down into granulocytes, neutrophils, basophils, eosinophils, and agranulocytes, lymphocytes, and monocytes. All right, the last of the formed elements is platelets, which are also known as thrombocytes. And they're not really cells, they're cell fragments that are produced in the bone marrow. So they're basically small fragments of marrow cells that take part in the mechanisms we call hemostasis. Hemostasis prevents excessive blood loss. Think of stasis means to stay put, and heme for blood. So since platelets are called thrombocytes, the production of them is thrombopoiesis, and it's stimulated by a hormone called thrombopoietin, which is secreted by the liver and the kidneys. Here's how it works. In the red bone marrow, hemopoietic stem cells with thrombopoietin receptors give rise to cells called megakaryoplasts. The megakaryoplasts then differentiate into megakaryocytes, which are extremely large cells that live in the bone marrow right next to blood vessels. The megakaryocytes have these finger-like extensions called proplatelets, which protrude into the blood vessels of the bone marrow and break off into the bloodstream. The proplatelets further break down into fragments called platelets, and that probably happens in the pulmonary vessels of the lungs. We store about 40% of our platelets in the spleen until our body's conditions warrant their release. Circulating platelets have a lifespan of up to 6 days or so. Since blood carries vital oxygen to our tissues, it's important that when a blood vessel ruptures, the rupture gets resolved before too much blood leaves the body. We call this process hemostasis, which is the cessation of bleeding. Obviously, when large blood vessels rupture, outside intervention may be necessary to stop the bleeding, but our hemostatic mechanisms are quite effective at closing breaks in the smaller blood vessels. There's a few hemostatic mechanisms, so let's take a look at each of them. The first thing that protects against blood loss is vascular spasm. It's a quick constriction of the broken blood vessel, decreasing its diameter and limiting the amount of blood that gets to the injury site. There are 3 initiators of vascular spasm. It can be stimulated by the triggering of nociceptors, which leads to vasoconstriction, or if the smooth muscle of the vessel is damaged, vasoconstriction could be directly initiated by those muscles. Platelets themselves release serotonin, which, as a chemical messenger, stimulates vasoconstriction. Vascular spasm only lasts a few minutes, but that's long enough for other mechanisms to kick in. The internal epithelial lining of blood vessels, called the endothelium, is coated with a slick, platelet-repelling substance called prostacyclin. So under normal circumstances, platelets don't stick to the endothelium. However, if a vessel is ruptured or lacerated, platelets contact exposed collagen fibers from the broken vessel wall. This causes the platelets to grow long, spiny pseudopods, that stick to the vessel as well as other platelets. Contraction of the pseudopods pulls the fragmented parts of the vessel's wall toward one another as the aggregated platelets form a plug to reduce or even stop minor bleeding. We call this a platelet plug. Platelets contain cytoplasmic granules that store and release biochemicals that also promote hemostasis. The exocytosis of these chemicals from the granules is called degranulation. And those chemicals are serotonin, adenosine diphosphate, and thromboxane A2. Serotonin is a vasoconstrictor that narrows the blood vessel's lumen to make the hole easier to plug. ADP, or adenosine diphosphate, attracts more platelets toward the rupture and stimulates their degranulation. And then thromboxane A2 stimulates the same events as the other two, which are platelet aggregation and degranulation, as well as vasoconstriction. Platelets are extremely important. You've got to be able to stop bleeding because we get little cuts all the time. Low levels of platelets is called thrombocytopenia and can lead to dangerous bleeding disorders. The third hemostatic mechanism that platelets are involved in is the formation of a blood clot, and that is called coagulation, which is a really involved process. So bear with me, I'm going to try to explain it in a way that makes sense in a podcast episode, but bear with me. It is pretty in-depth and there's a lot of different biochemicals to keep track of. So once again, that formation of a blood clot is called coagulation, and it's the most effective hemostatic mechanism and the last step of stopping blood loss. Sometimes you'll hear this called the coagulation cascade because of the series of events that are required to make it work. The coagulation cascade involves over 30 chemical reactions to make sure we don't lose too much blood and that a clot can quickly occur when a vessel is broken. The objective of coagulation is fairly simple. Convert an inactive plasma protein called fibrinogen into its active form, fibrin. Since fibrin is, in general, a sticky protein, it sticks to the walls of the blood vessel. Then blood cells and platelets stick to the fibrin as they flow past the site of vessel damage. This mass of fibrin, blood cells, and platelets is called a blood clot, and it seals the ruptured blood vessel. It seems really simple, but the formation of fibrin is actually very complex. There are two chemical reaction pathways to coagulation, the intrinsic and extrinsic mechanisms. The intrinsic mechanism is initiated by factors in the blood itself, which is why it's called intrinsic, while the extrinsic mechanism is initiated by factors coming from the blood vessel or the structure surrounding it, which is why it's called extrinsic. Clotting factors, called procoagulants, are usually inactive proteins, most of which are produced by the liver. Once one factor is activated, it actually functions as an enzyme that activates another factor. Then that factor activates the next factor, and so on and so on. It's a domino-like sequence of events called a reaction cascade. When you read about this, you'll notice that we use Roman numerals to identify each clotting factor, but they're not numbered in the sequential order they act to achieve coagulation. I know, it seems like, why would they do that? Why are they trying to confuse us? But the reason they're numbered the way they are is because they're numbered in the order in which they were discovered. So be careful not to confuse that. You're also going to notice that some Roman numerals seem skipped. That's because after they were initially discovered, it was found those factors were redundant and eliminated from the nomenclature. Some clotting factors are produced by the platelets and called platelet factors. We're going to discuss platelet factors 1 through 4. Okay, so coagulation is divided into two phases, initiation and completion. The goal of initiation is the activation of what we call factor 10 with a Roman numeral. The extrinsic and intrinsic mechanisms are two different methods of initiation, right? So there are two different methods of getting to that factor 10. Once factor 10 is activated, the completion phase begins, which is the same whether factor 10 was activated by the extrinsic or the intrinsic mechanism. Let's look at the extrinsic mechanism first. When a tissue is injured, it and the ruptured blood vessel release factor 3, which is a lipoprotein called tissue thromboplastin. In the presence of calcium ions, factor 3 combines with another procoagulant called proconvertin, or factor 7. The complex they form activates factor 10. So the extrinsic is pretty simple. The factors that drive the intrinsic mechanism are found in the platelets and the blood plasma. The platelets granules release factor 12, which activates factor 11. Activated factor 11 activates factor 9. If calcium ions and platelet factor 3 are present, they assist factor 9 in activating factor 8, which is also known as anti-hemophiliac factor A. A genetic inability to synthesize factor 8 is the cause of classic hemophilia, which is a dangerous condition that makes it difficult to form blood clots. For people with hemophilia, minor injuries can lead to extreme blood loss. This is a genetic condition where you don't have the DNA sequences on the gene to form a proper version of the protein for factor 8. And then factor 8 activates factor 10. Now that factor 10 is activated, let's discuss the completion phase. If calcium ions and platelet factor 3 are present, factor 10 combines with factors 3 and 5 to produce prothrombin activator, an enzyme that converts prothrombin, which is factor 2, to the enzyme thrombin. Thrombin then converts fibrinogen into shorter strands of fibrin monomer, which bind to each other to form longer fibers of fibrin monomer, which bind to each other to form longer fibers of fibrin polymer. Active thrombin also combines with factor 5 to produce more prothrombin activator, which in turn leads to more active thrombin, making this a self-amplifying positive feedback system. Factor 13, also known as fibrin stabilizing factor, connects the fibrin polymer strands from across to create a mesh-like web that can catch blood cells and platelets as they flow into it. The combination of the fibrin framework and the blood cells stuck to it forms the blood clot. Each step in this process produces a multitude of enzymes that activate the next step in the sequence, amplifying the process as it moves forward. This is called the reaction cascade of blood clotting and results in very quick production of millions of fibrin molecules. And finally, extensions of the platelets called pseudopods stick to the fibrin and contract, pulling the framework toward the center point and drawing the damaged ends of the vessel together. Now, this is all really important when you have an injury. However, forming blood clots too readily, when it's not necessary, can be really dangerous, because these blood clots can grow to a size that can block arteries. So if you've got an artery in your heart, for example, that is a little bit narrowed because of atherosclerosis, which means plaque formation underneath the endothelium has formed as a result of high cholesterol, high blood pressure, genetics, diet, all these kinds of things, saturated fats, that can make the blood vessel narrower than it normally is, which means a blood clot that normally would pass right through it with no problem will block it and prevent blood from getting to the part of the heart that it's supposed to supply. That is extremely dangerous, and that is going to cause what's called ischemia, which is a low blood flow, which leads to hypoxia, which is low oxygen levels, which leads to an infarction, which is a death of the tissue. When it's the heart, it's a myocardial infarction, or MI, which we commonly call a heart attack. So people who are at risk for these things can sometimes take anticoagulant drugs like heparin and Coumadin. Plavix is another one. And that will decrease the likelihood of blood clots to form to protect from catastrophic outcome. Now, the downside is that you got to watch your bleeding time and you got to be careful, because you don't want to excessively bleed because you don't have the coagulation abilities now because you're taking the drugs. It's a delicate balance, right? It's a delicate balance to make sure someone is staying alive, staying healthy, but also the side effects are not making them unhealthy.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times. But we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
All right, that is a lot. We finished up blood right there. We talked a lot about leukocytes, about platelets, about coagulation, leukopiosis. That's a lot to do in one episode. So I know that that was a big chunk. I hope that that information and the way I've explained it is helping you get your B or better in A&P. So I'm going to sign off now and say good luck on your next A&P exam. I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media. Please take the time to rate the podcast, and don't forget to check out my YouTube channel, Student Help for AP. That's Student Help, the number 4, AP. There's a whole lot of tutor videos on there that I think you're going to find helpful. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where you can find Anatomy and Physiology Digital Suite, my low-cost, tutor video-based digital learning solution for anatomy and physiology, already being used at several colleges and universities.